Development and success of adoptive cell therapies in solid tumors is dependent on innovations that will address challenges like heterogeneous nature of the antigens on tumor cells, antigen escape, lack of tumor specific antigens that can be targeted, uncontrolled activation of immune system, immune suppression caused by tumor microenvironment, and low infiltration of the tumors by these cell therapies and the low persistence. Several strategies have been applied to engineer the cell therapies to overcome the various challenges. For example, use of suicide switches, conditionally activated and logic gated cell therapies, or use of small molecules to regulate CARs. These improve their safety profile. Many companies are engineering cell therapies to address limited cell proliferation by constitutive expression of cytokines or the synthetic receptors.
After CAR T cells, now let us move ahead towards understanding the current technology and innovations that are being considered for personalizing TCR-based therapy to address unique aspects associated to the diverse solid tumor landscape. Now, the technology advancement is such that the development of T-cell receptor isolation technology and genetic engineering programs have enabled patients' T-cells to express TCRs, recognizing multiple peptide and HLA combinations to specifically recognize tumor-associated antigens and to effectively eliminate tumor cells. So let's go on to our first platform, that is Adaptimmune's specific peptide enhanced affinity receptor SPEAR T-cells, which focuses on affinity enhancement to enable improved antigen binding and reduced off-target toxicity. Next, we move on to Sleeping Beauty platform developed by Allonos Therapeutics, which targets shared hotspot mutation through T-cell therapy library. Now, Sleeping Beauty achieves this by non-viral gene transfer by genetically modifying the patient's T-cells, wherein the patients are screened to confirm the tumor mutation HLA through sequencing. The information is then utilized to screen the patient for a match to the TCR library, and a pre-manufactured TCR DNA plasmid is selected, making this process quick, efficient, and highly personalized.
Summarizing the key developments, innovations in cell therapy design such as improvements in persistence, functionality, expansion, safety, as well as allogenic approaches, coupled with advancements in manufacturing processes such as reduction in vein-to-vein time through improved gene transfer protocols, in vivo expansion, use of younger phenotype cells, are steering the cell therapies towards success even in solid tumors. Expected approval of lifileucel further validates the potential. Increasing decentralization of trials, exploration of cell therapy administration in outpatient settings, and guidance from real-world data will support better patient access, adoption, and regulation of the cost of these therapies.